Hi, this is Miss Litton, and this is lunchtime on a Friday, and I have a few powerful AP biologists that showed up for their review today. And we're on Enduring Understanding 1C, and that big topic that you would want to be able to support on your AP exam is that life continues to evolve within a changing environment. So the first thing I want to talk to you about is the difference between macroevolution and microevolution. Whoever's sitting in a blue seat, tell the slated person, what is the difference between macro and microevolution? Which one is within a species? Microevolution. And changing to a new species? Macroevolution. Perfect. Okay. That's really funny. You should be laughing really hard at this.
Wait, no,
Okay, never mind. All right. So, speciation, and you're taking one and maybe splitting it into two lineages, or one is changing into another. We talked about what a species is because that part's kind of tricky. Because remember, species is a group of organisms that are interbreeding. And sometimes neighboring groups will interbreed, but then you get two neighboring groups away and they won't interbreed. And at what point do you call it a new species? And is it that they're not interbreeding because they just don't have access to each other? Are they not interbreeding because they wouldn't know how to interbreed, or their parts don't fit together? So the key thing here when you talk about a species is that they're interbreeding and they're sharing a gene pool. And the types of things you can look at, you can look, and I'm sorry this is overlapping here, you can look at the DNA and look to see how similar it is. We've talked about that before. You can't, can anybody read that through there? You know what that says? Perfect. DNA hybridization techniques. Now, if I told you something is a hybrid, what would that probably mean? Yeah, mix. So if I'm doing DNA hybridization, I'm taking one strand of yours and trying to see if it matches, if it's complementary to another strand of a different species. The more they line up, right, the more similar they are. So those are some of the things that you can look at. As far as, why isn't the slated person? You tell them this instead of me. You speak it to them. Go ahead, slated person.
A species is a Latin word meaning.
Okay, more than one of you should be telling this to your neighbor. Here you go. Own it.
Differences that are used to distinguish species. Today, differences in body functions, biochemistry, behavior, and genetic makeup are also used to differentiate.
What would be hard, looking at this definition, what would make it difficult if you're looking at a fossil? What would be difficult if you were looking at a fossil? Say. What?
Genetic evidence, looking at the biochemistry.
I can't even hear you, so you're
Looking at the biochemistry.
looking at the biochemistry. Yeah, would there ever be any biochemistry I could look at in a fossil?
no.
Maybe something preserved in amber, perhaps? Right? If the whole organism is preserved, like a woolly mammoth in ice, would I still be able to look at his DNA? Yes. What else on here would be difficult? Behavior, exactly. The way an organism acts or responds, you would have a difficult time quantifying that from a fossil, right? Okay. So that's part of the struggle when you look at those aspects of speciation. This is going through, remember we said there was the different definitions of a species, if you remember that from the presentation. So biological is looking at reproductive isolation. Morphological, you're looking at anatomical differences. However, if you compare a placental mouse to a marsupial mouse, they might look very, very similar to you, and it would be hard to differentiate, or like a flying squirrel. So you have to look, again, probably your best bet is to go and look at the DNA. So then we talked about isolating mechanisms. What would keep species separate? And so on the reproductive isolating mechanisms, it could be something that's structural, it could be a function, it could be a behavioral characteristic that keeps them reproductively isolated. And we looked that there were pre-zygotic isolating mechanisms, like before a zygote ever formed, and those are probably more common. And then if there's pre-zygotic, there must be what? Post. So do you see how that's very wasteful if your isolating mechanism happens after you've already invested in an offspring? That would be post-zygotic. So here are some of the different ones, and why don't you just ping pong back and forth with somebody that you're next to. Go through these first three, and then I'll switch the slide. Better if you review it yourself and kind of put it in your own words than I say it for you. Go ahead.
So, geographic isolation so nature is separated into species and they cannot interbreed anymore, and does that cause changes?
So ecological isolations, they occur in the same area, but they have different places of that same area, so they don't,
We good on these? Okay, so on the first one, right, you are, there's no way you're gonna meet. This is like trying to date somebody in New York right now, right? Okay, it's just you're not gonna run into each other. That would be geographical isolation. Or like, never mind, because it has to be different species. And then ecological isolation, you're right, you're living in the same exact community, but you still, there's slight differences. You're on the top floor, they're on the bottom floor, whatever. It's something to keep you separate. And then temporal would be you live in the exact same place in the same habitat, but you don't want to reproduce at the same time. So there's some sort of temporal isolation. Okay, keep going. Here's 4 more, including post-zygotic. Go ahead.
So behavior isolation is how they work, what they do. Gamete fusion, prevention of fusion.
And then you have post-zygotic.
Now, the sooner the isolation steps in, do you see how the less wasteful it is? Like if you're waiting and it's this part, you get all the way to this point. Okay, if your pollen has already grown a pollen tube down into that flower, and at the very end you can't get the 2 gametes together. Do you remember how we talked about how when we talked about development and how the sperm binds to a receptor on the female? So if you get to the point where you've done all the parts of reproduction, and at the very end your sperm can't fuse with a receptor on the egg, that's very wasteful. So the earlier on you have your isolation, the better. Here, you're actually having a zygote is developing, you're spending the nutrients of the mother nurturing, you're maybe even feeding it milk if you're a mammal, and again, if that's not going to go to fruition, girls in the back, then that would be, very wasteful. Okay, so here are those pictures. I think that was a good summary, so this should hopefully make sense. The only reason I want to show you a few of these is if you had to use them for an example. So all these frogs live in the exact same community, same habitat, live in the same regions, but their mating activity is just slightly off one another, so that maintains their reproductive isolation. Behavior, do you know the dance? Mechanical, do your parts fit together? Okay, and so I think this is all going to be post-zygotic, so after you've formed a zygote, it starts to develop. Sometimes the F1, do you know what I mean by F1? The first generation of offspring? They are actually viable and can mate, but by the time they make offspring, which would be an F2, they are not. Okay? All right. So the 3 that I just talked about, those are right there. Why doesn't the oldest person, whoever's sitting next to you, decide who's oldest? Just run through these real quick, or assign them to your younger partner, whichever you want to do.
Okay, reduced hybrid viability, genetic incompatibility between the 2 species may thwart the development of the hybrid at some embryonic stage and produce frail offspring. Reduced hybrid fertility, even if the hybrid offspring.
All right. Can I move on? Okay. So this was an example of zygote mortality. These are 2 different species, and they can mate. The young starts to develop, but then it would be like a miscarriage. It wouldn't go all the way to fruition. Okay. Here you have a sterile hybrid. And here they have reduced fertility in the F2 generation. All right, so then we said, what are some of the causes of speciation? The most common cause would be allopatric speciation. Some, like a river runs through it, there's some sort of barrier, you diverge, you go down your own pathways, you have your own, what I'm trying to do right now? Mutations. You have your own selection pressures, you have your own initial gene frequencies, right? Remember how we talked about what you start out with? Remember when we talked about genetic drift? Remember the examples of genetic drift? What are the kinds there? Genetic drift. Bottleneck, you go through a crisis, who makes it to the other side? And what was the other one?
Founders.
Founder's effect. So this would be like Founder's effect. So when you diverge, if I say half the people over here are going to go and start a whole new population, and this half is going to go and start a whole new population, look at the variations between the two halves of the room, right? We're all humans, but there are differences amongst us. So that initial gene frequency can be different. You can experience different selection pressures. Their island might be more desert-like. Our island over here might be a tropical rainforest with beautiful beaches, okay? And then we undergo different mutations. It's not going to be the same, and the mutation is going to change as well. So that's allopatric speciation. That is the most common form. Some sort of geographic barrier comes between you. Okay, why doesn't oldest bio buddy explain the one on the left and youngest bio buddy explain the one on the right? And what other type of speciation is getting shown here? Go ahead.
So the left one is talking about allopatric, so it's a geographic barrier, companion or river, two species.
Okay, do you remember the other form of speciation, what that one's called? Sympatric. And usually that has to do with what? Chromosomes, something, right? Something with the DNA, because there's something that changed here, some sort of mutation, some sort of doubling down of chromosomes or something. And this is more likely to happen in plants than animals that cause that change. Okay, so here's allopatric speciation starting with a barrier. A good example of that is salamanders traveling from Northern California to Southern California. The barrier they ran into is, the desert. So some went left, some went right. They accrued differences, the different initial gene frequency, different selection pressure, different mutations. So even though they're rejoined here at the bottom, they are totally different species at this point. These are squirrels on either side of the Grand Canyon. So the Grand Canyon wasn't always so grand. So at first it was just a river running through it, and as they separated, then we have, you can see just even the environments they live in are different. Why doesn't, there's something new in this slide than what I've said before. Why doesn't the youngest bio buddy take it on? Go ahead. What do you see that's new in this slide? So what's new in this one? What do you see that's different than what we discussed so far?
They come back together.
They came back together. So just because you have, girls, this geographic barrier that separates you, okay, you might have accrued some differences, but if you interbreed ultimately and you kind of erase away those differences, then speciation is not occurred. Okay, and sympatric speciation we already talked about. It's happening, they're right here in one area and something has changed. Usually it has to do with chromosome numbers. And again, this works for plants, does not work for animals usually. So in this case, their haploid number was 14 for the wild wheat species, and this other species, the haploid number here is 7. And when they get together, their chromosomes don't line up. So you would think hybrid, in viability. But all the chromosomes underwent mitosis, so now they have 2 copies of everything, and they're now diploid again. They can now undergo meiosis and form eggs, and then that would be like today's wheat bread. Usually, polyploids, when they have extra sets of chromosomes, the fruit is bigger, everything you want, and so we have a lot of polyploids that we eat today. Okay, then we talked about 3 types of selection. Here is the first one. See if you can remember the 3 types of selection and then identify which of those would lead to speciation. Go ahead, have that discussion.
Directional, disruptive.
Starts with an S. Stable. Yeah, stabilizing, yeah. Okay, what's this one right here? Directional, right? Oh, sorry, it's on the board. Sorry. What's this one? I don't know, Miss Liz. What is it? Read it. This is found in a good example. That is a horse. As the horse's environment changed, then what was adaptive changed, right? And so as they had less trees, forest areas to live in, and they had more open fields, then it was better if they were taller so they could see the predators coming. So this is a good example of directional selection, and that would lead to speciation. Obviously, this big guy and one of these did not interbreed well. Okay, stabilizing selection. It's not too hot, it's not too cold, it's just right. Thank you. And would this lead to speciation? No, you're staying constant, uniform. So it's kind of like the black ones don't work, the white ones don't work, you need the gray, right? Okay. All right, and disruptive speciation. This is where you favor the 2 extremes. Would this lead to speciation? Yes. Okay. All right, this is just a statement. Oldest bio buddy, take it on. And that would be because speciation is the process of what? Process of forming a new species. Good. So you're in between. All right. I wanted just to show you, this is a picture showing you that hybrid zone. If there's a few in there, they can gain ascendancy, and if the hybrids work, this hybrid could be a new species, potentially, or it might just bring you right back to your original species. Okay, youngest bio buddy, this is you. Go.
Okay, this is what the
Okay, and oldest bio buddy, go ahead.
When closely related species meet in a hybrid zone, there are 3 possible outcomes. Strengthening of reproductive barriers, weakening of strengthening barriers, and information coverage.
So it either reinforces your differences, right? Or it starts to smooth over your differences, or you might become a new species. You might end up with 3. Okay? All right, so then we talked about sexual selection, and there was intersexual selection and intrasexual selection. Intra is within, like intracellular environment is within a cell. So when you look here, this is Joe working against Jim and vice versa. They're the same, the 2 male working, and one is trying to outcompete the other. Intersexual selection is when, in this case, Betty is choosing either Joe or Jim. So if you think about it from the male's point of view, they have to compete, right, with each other to even be in the selection, to even be looked at. And then on top of that, they still have to have the female choose them. So when they're competing with each other, could it be a fight? Sure. Could it just be the colors, their song, their dance? Yes. So they may not be competing directly with each other and coming in contact with each other, but the female's going around and looking, and she's looking for one maybe that has the brightest colors or whatever it is. Now, this intrasexual selection can also lead to differences between the way the males and the females look. Okay, so this is male fighting male in intrasexual, and then intersexual. This is where we get. Do you remember what that word is called? Is it already on the board? No. Sexual starts with a D. Dimorphism, where di means what? 2, and there's 2. Depending on your sexuality, you're either a male or a female, and you look different from the other. And we could talk about why is it that the females tend to look plain. Not that they're doing the choosing.
They're not attracting females.
No, it's not that they're not attracting the females. I'm looking for something else. Why else would a female want to be plain?
They don't need to compete.
No. Think defensive mechanisms.
Oh, so they can defend them. So that they're not like found by predators?
Yes, they're not found by predators. If they're the ones laying the eggs, they should be plain so they don't stand out. They don't want to be brightly colored. Okay? They leave that the male to go peacocking around. But the female wants to look, reduced. She doesn't want anybody to see her. All right, and then we talked about adaptive radiation. And this is sometimes following some sort of extinction event. And this is where you have a whole bunch of vacancies, and you're just like, I'll take the top bunk, I'll take the bottom bunk. You're dividing up the regions. And exploiting then those aspects of your morphology that would survive in that particular level of the forest, or that tree level, or the type of food that you're choosing. It's very rapid, very competitive, and then you start to accrue those differences where some have bigger beaks or some have smaller beaks, etc. All right, and we talk about Darwin's finches, but an even better one is the Hawaiian honey creepers. You can see huge variations in their beaks right here. All right, and then a really good example, and I know you've seen this if you took a practice test, yeah, that always comes up is that damn stickleback, okay? And they always use it, they study it, it comes up, it had that really, I don't know if it was in this test or I gave you a test one time with a bunch of genes and you had to compare it. Was that an AP exam or was that a real test? Yeah, they'll talk about the stickleback, and so just grow to love the stickleback. And so on this one, they'll talk about the lateral plates on it, and the saltwater oceanic ones have more lateral plates than the freshwater. There's a few genes they look at, and they trace these few genes and make comparisons between them and the base sequences between them. Depending on their age, they can have variations in plates. Depending on what type of water they have, they can have variations in plates. So you just want to be like, okay, stickleback, I'm cool with that. So if something comes up, just be like, I love the stickleback. Ask me a question about it. Okay, and then I don't know how many questions I have here, but I'll give it a go. And I don't know if they're good questions. I did not look at them again. Be your favorite thing to do this weekend that I would want to hear about. Be the favorite thing you like to do this weekend that I would care to hear about.
So for adults,
You
You do Ross job? Okay. You do it. Not that that's any of my business. Ooh, somebody's doing boot camp. Oh, Ryan Gosling. I'm in. I'm assuming you're referring to the movies.
Sure, yeah.
Do your Amgen lab, do sleep, doing biology. I'm going to still go with Ryan Gosling. No offense, Ross John.
thank you.
Here, you got 5 questions. I'm going to pause this for a minute while you do those. And Ryan Gosling. Okay, here we go. Ready? In. Get your score.
I told you. You have no idea what
Yes.
10 out of 10.
Have I taught, did I review phyletic gradualism and punctuated equilibrium?
Yeah.
Punctuated equilibrium is rapid change, like over maybe 1,000,000 years, and phyletic gradualism is just like it sounds, gradual. So I think punctuated. Okay, and then when only a few individuals survive unfavorable times, thereby lose the majority of phenotypes in the next generation, it is called what? Bottleneck.
True.
You all got that? You all got that? You all got that? Yay! I'm so proud of you. Good job, you guys, for coming in. Well done.